The next presentation is given by Gabrielle Martin, who works at the University of Toulouse and who has been working on the monitoring program Vigiflor for quite a few years now. She did a PhD working on Vigiflor. Thank you, Gabrielle.
Thank you, Emmanuel, to offer me the possibility to present the Vigiflor monitoring scheme, and thank you to the Collège de France team. So, yes, I'm working in Toulouse, but also, namely, I'm working on this monitoring scheme with the team at the Museum of Natural History in Paris. And I'm going to present you this program called Vigiflor, which is a French monitoring scheme of wild flora in France. So for a long time, the aim of species monitoring has been to assess locally the effects of human pressure on rare species, and mainly for practical reasons, as for example, because these species present reduced distribution area and small populations, and so the effects of pressure are more easily perceived in this case. However, rare or endangered species represent a small proportion of the total species biomass, as opposed to most common species. And these common species are characterized by a wide distribution range, namely they occupy several types of habitats, and at least they represent one large population in some case. They represent a significant proportion of total biomass, and they play a central role in the functioning of ecosystems. So in this framework, the variation in abundance are less perceptible for this species, and we need for large amount of spatially distributed data to detect them. Standardized long-term monitoring biodiversity offers the possibility of comparing biodiversity in space and time, while avoiding most of the biases associated with opportunistic data, and particularly when the sites monitored constitute a representative sample of ecosystem. These standardized long-term biodiversity monitoring programs, which are based on a network of permanent sites, for example, plots or transects, a representative sample of the territory or habitat based on random or systematic sampling, based also on repeated visits to fixed sites, and on a standardized protocol to collect presence and abundance data or species frequencies, are essential in ecology and make it possible to account for subtle temporal change in population, species, and communities. In France, the Vigiflor standardized wild flora monitoring scheme was launched in 2009, and it offers to amateur and professional botanists the possibility to get involved in observing plant communities in all types of habitats. The participants are considered as volunteers for their implications, and the objectives of these programs are to detect, measure, and understand changes of common flora, namely by calculating or by assess temporal trends of common species, to detect some decrease, increase, or increase of species frequency, to calculate changes in plant community compositions. Another aim is to detect relations between changes and human or environmental factors. And the third one is to produce biodiversity indicators as for example, we work on species or multi-species frequency indicators, on species richness or diversity and beta diversity indices, on functional traits or ecological preferences, and on ecological functions, as for example the eutrophication or food for insects. I will present some results on some subjects. This monitoring of wild flora in France, called Vigiflore, has been set up according to several principles. The first one was to retrieve a representative sampling of the territory. So the sites are selected on a systematic grid. The second principle is to develop a standardized protocol to allow comparisons in space and time. The third one is to collect the presence and frequency data as a proxy of abundance data. And the last one is to call for contribution from volunteer botanists in France. So there is a standardized protocol which has been proposed to enable each participant to collect the data in the same way. And here is a representation of its standardized protocol. So you can see a square, which is the sampling unit of the Vigiflore program, and which is corresponding to a square of 1 kilometer per side. And each square has been predefined in France in a random and systematic way. So each square is separated from another one from 10 kilometers in France. And all these squares are offered to volunteers. So within each square, you can see there is eight circles, and they represent 8 plots of 10 meters squares, which have been systematically and randomly defined also. And we suggest to participants to visit each plot in a square and to sample flora in an exhaustive way. So here is represented the map of the 715 Vigiflore squares sampled since 2009, and there is more than 400 observers in the programs since the beginning of this program. And all these squares, so in all these squares, it is 3,008, more than 3,800 plots which were visited at least once or more frequently during the period. In each fixed plot, observers can also collect some environmental variables. As for example, they can precise the slope, the shadows, soil type, habitat according to the current biotope classifications, or signs of degradations of the plant community or the habitat. The idea is to identify all plant species in each quadrant in a plot, so we can calculate the frequency of a species with a number of quadrats for each species. For this program, there is a network of observers, and we share scientific results and mutual support. There are some observers who are agree to help other observers, and there are 14 regional referents who suggest to help with setting up the protocol, help to species identification or checking data, for example. And each year we organize national and regional meetings in France, in different regions each year, to mobilize the network of observers. And to introduce you some numbers about this monitoring scheme. We celebrated the 15 years of the program last year, and it was more than 150,000 observations which have been sent by observers, and they represent more than 13,000 records. And we observers identify more than 2,600 species belonging to 150 botanical families. And in the database, we have 90% of observation identified at the specific level. So there are there are a pool of most sampled species, which represents the most common species. And we have lots of species in the database which have been observed only once since 2009. And here there is 3 examples. So to give you some other numbers about this monitoring scheme of white flora in France, before to introduce the important results, we detect that the squares and plots are monitored for an average of between 3 to 4 years, and there are 5 plots which are sampled per square on an average on 8. Some environments are underrepresented in relation to the surface areas they occupy on a national scale, and this is the case for agricultural areas, while others are overrepresented, and this is the case of urban areas. So we explain that by the way that observers mainly live in cities, and when they choose a square not so far from their home. So the probability for the squares and for the plots to be in urban areas is important. And it is the map which is represented here. The color represents the number of years of monitoring per square, and you can see that it's not homogeneously distributed in all France. In the Paris region, we have lots of some squares monitors a long time than in other parts of France. To speak about analysis, we perform some analysis at the species level and at the community level, namely. So at the species level, we focus namely on to calculate species temporal trends, and at the community level, we worked a lot on plant functional trait and community weight and mean traits. And we take namely into account life history traits of species, for example, as life cycle, thermal preference of species, dependence on pollinators, and other floral traits. So this is an example of some species for which we assess the temporal trends. So we can assess the species temporal trend for more than 600 species, which represent the more common species in the database. And they represent, to give you an idea, they represent around 80% of the observations in the database, all these species. And you can see that there are lots of different patterns, namely some species increasing and some decrease, and also some stable temporal trends. We focus a work on response of wild flora to climate change, and for this, the data collected revealed rapid changes in the composition of plant community in relation to climate change. So we looked at the thermal preference of plant communities in France using the Hillenberg indicator, and we calculated the average thermal preference of plant communities. We then looked at how this average thermal preference of plant communities varied over time. And this is what is shown in the graph. We showed a significant increase in the index over time, which revealed a recomposition of plant communities in response to climate change on a national scale in France over a short period of time, as you can see. And we detect the same signal at the regional scale for the Parisian region, namely. And to understand what this significant change was related to, we looked at the temporal trends of the most common species in France. And here is the phylogeny of the 600 most common species in France, for example. So for each of these species, we calculated the species temporal trends to detect increases, decreases, or stable trends. And we look at which species were declining and which were increasing. And we found that declining species are shown here on the graph in the right in red, and increasing species are represented in blue. The longer of the bar is proportionate to the slope of the temporal trend. And we were able to show that the increasing species are species with higher thermal preference on average, whereas declining species are species with a low thermal preference on average. So this reshuffling of plant communities, therefore, seems to be linked to an increase and to an increasing abundance of the species best adapted to high temperature. And this result is particularly marked for annual species in our dataset. Annual species with a high thermal preference tend to increase more than species with low thermal preference. And I put two examples. The example of the Avena barbata, a Mediterranean poacea, which is increasing in France in abundance, in frequency in communities. And I can show you, I can present you a second work. So in addition, we performed some analysis to try to detect relationship between species, temporal trends, and pollination syndrome. And the first step was to identify pollinator dependence of each species. And so this graph represents the number of common species according to their pollinator dependence. And we can see that there is an important amount of species independent to pollinators for their reproduction. There is a second important amount of species totally dependent to pollinator for their reproduction, and there is a third group of species dependent to pollinator in various ways. And we highlight that we detect an average increase in all pollinator-independent species versus an average decline in all plants totally dependent on pollinators in France. To further understanding these joint temporal variations in plants and insects, Solène Agnew, which is working on the Vigiflor database, she's a PhD student at the National Museum of Natural History, is working with the Vigiflor dataset, namely in association to other datasets, to understand what are the characteristics of declining or increasing plant species, what are the consequences of changes in flora on the floral resource availability, namely nectar and pollen for pollinating insects, and what are the relationships between temporal trends in plants and temporal trends in insects in relation to global change. So Solène is working on the production of indicators also, and one indicator could be to map the resource availability in space and time for pollinators. So for this part of work, we are working on floral traits to try to identify more precisely pollination syndrome and relationship between species temporal trends and pollination syndrome. So we focus on flower color, for example, flower shape, flower symmetry, the flowering period, the presence of UV reflection pattern, etc. And we want to explore the question, what are the consequences of changes in plant communities on the resource availability for pollinating insects. To conclude, this is my last diapositive. So these data collected in the Vigiflor framework are collected in a standardized way, but are they free from bias? So I didn't put these results of this analysis here, but we detect that there is trends in the behavior of observers. We have an important participant turnover in the network, and the squares with the highest species richness are monitored for longer period than the others. Considering the data quality, the regional reference help us to increase, to work on this point. And after we performed some analysis on the sampling pressure required to detect changes in plant communities. Voilà. Thank you to the Vigiflor team and to all the volunteers in France.